Ladies and gentlemen, welcome to Geotalks Online. Thank you for joining us. We have 40 attendees already, so thank you for everyone who's joined us today. We must, of course, thank John Hancock's and CCIC for sponsoring these Geotalk Online events, and of course the School of Geosciences. As you may have remembered if you joined us last week, we have a Q&A functionality on Teams Online, and you'll notice that there's a Q&A bar on the side of your screen. You can ask questions at any time during the talk, and at the end of the talk we'll publish those questions one by one, and we'll deal with the questions then. But you can write those questions and submit them at any time during the talk, so please feel free to do that. We'll also post some links to some of the papers that the speaker will talk about today in that Q&A bar on the right-hand side of your screen. So today we have a fantastic talk by a recently graduated Wits student, or no longer a student, now a postdoc, and that is Kimberly Chapelle, and she joins us from the Evolutionary Studies Institute at Wits University. And Kimmi did her undergrad studies at Wits and also completed a PhD at Wits last year, working with several supervisors, but mainly Professor Jonah Chonier. And the focus of today's talk is on her PhD studies on Massospondylus, and she'll tell us all about that as the talk gets going. She is obviously funded by the Evolutionary Studies Institute and is doing a postdoc through the Center of Excellence in Paleosciences. And next year, we'll take up a postdoctoral fellowship at the American Museum of Natural History. So thank you very much, Kimmi. I'm going to load up your video now, so you will see Kimberly fairly soon. Kimberly, you can unmute yourself if possible.
Hello.
Here we go. So Kimberly is now live. Over to you, Kim. Enjoy the talk.
All right. Thank you everyone for joining, and thank you Grant and everyone for organizing this. I'm very, very happy to be talking to you today. This is the most social interaction I feel like I've had in about 5 weeks. So thank you. And as Grant said, today we're going to be talking about my PhD research, which is all about Massospondylus carinatus. If you don't know who that is, don't you worry, I'll introduce you to him. And more specifically, how did Massospondylus grow? Yeah, before we get into that, I get asked a question. For some reason, my PowerPoint won't go now. This always happens. All right, so one of the things I often get asked is, why study dinosaurs? What is so interesting about dinosaurs? And there are many, 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 many reasons for which dinosaurs are really important and really interesting to study. I'm only going to go through a few of them. One of those reasons is that dinosaurs appeared approximately 240 million years ago, which is a very long time. And they were the dominant life forms on Earth until the end Cretaceous extinction, which was about 66 million years ago. So that means that they were dominant for about 170 million years. That's an enormous amount of time. And if we think about it, birds today are related to dinosaurs. They evolved from meat-eating dinosaurs, theropods, and they actually survived that end Cretaceous extinction. So their dinosaurs are still around today. Another reason is that dinosaurs during those 240 million years managed to survive 2 major extinction events. So the end Triassic extinction, which was about 200 million years ago, wiped out approximately 50% of all of life's Earth's life, and dinosaurs made it through that. The end Cretaceous extinction wiped out about 75% of all life on Earth, and dinosaurs made it through that, and we still have birds today. So dinosaurs are extremely successful, a very successful group of animals. They're also a very diverse group of animals, and they've managed to overcome and come up with all sorts of different feats in engineering. As well as play around with their physiology quite a bit. So we have things like sauropods, which are the largest animals to have ever lived on land. And that comes with a lot of adaptations, such as, how do you keep your head up without all of the blood, draining. So there's a lot of physiological adaptations with that. They evolved powered flight, they evolved body armor that play around with body proportions. They're just an extremely diverse group of animals. And in fact, they still are a very diverse group of animals. If we look at birds today, there's over 10,000 species of birds on Earth today. There's only about 5,000 species of mammals. So they are just an excessively successful group of animals. And I know that this might sound very cheesy, but that's all right. You can't know where we're going. We can't study the future. We can only predict the future. And in order to make predictions, we need to understand what's happened in the past. And dinosaurs really are an excellent way of studying the past and understanding how ecosystems react. Okay, so now we know why we should study dinosaurs. Something that a lot of South Africans specifically don't know is the amazing fossil record and dinosaur record that we have right here. And one of those really important species in taxon is Massospondylus carinatus. Now I'm slightly biased. I think Massospondylus is the best dinosaur on Earth. That's because I've been working on it for about 8 years. But it is honestly a very important dinosaur, and I'm going to tell you why. So Massospondylus, first of all, was named in 1854 by Sir Richard Owen, which is very early on in dinosaur descriptions. The very first dinosaur to be officially named was in the mid-1820s. So Massospondylus, which is a South African dinosaur, was actually one of the first dinosaurs named. It's a Southern African basal sauropodomorph dinosaur. For those of you who don't know what sauropodomorph dinosaurs are, it's a large group of plant-eating dinosaurs that includes things like the big graviportal sauropods that we all know and love, like Diplodocus and Brachiosaurus. But those guys started off as much more humble bipedal animals like Massospondylus. So it's about 5 meters long from head to tip to tail. It walked on 2 legs, it ate plants, it had a very, very small head. But that's all right. And it is the most common dinosaur that we have in South Africa. It's only found in South Africa, Lesotho, and Zimbabwe, so it's a very proudly South African dinosaur, and we have hundreds of them. It lived in the late Triassic to early Jurassic time period, so that's about 200 million years ago. For all the geologists out there, I am not a geologist. I have very talented colleagues that do this stuff for me, with me rather. And so because it lived in the late Triassic to early Jurassic, that means that we find it in what is called the Upper Elliott and Lower Clarence formations. So that's part of the Stormberg group. It's mainly around and in Lesotho. So we work a lot in the Free State and the Eastern Cape. That's where we do most of our digging. Because it lived as it stands from the late Triassic to the early Jurassic, that means that Massospondylus would have been around during the end-Triassic extinction. The end-Triassic extinction is one of the big 5 extinctions. As I said, it wiped out over 50% of all life on Earth, and Massospondylus seems to have made it through that. So Massospondylus becomes an extremely important taxon to understand how species recover in post-extinction environments. So even though Massospondylus was named a very long time ago in 1854, surprisingly, it wasn't described in detail for a very, very long time. There were papers published on it, but because it resembles other dinosaurs in its family, it wasn't described. Its skull and its anatomy weren't described in very high detail until very recently. And one of the things, one of the technologies that really helped me and us with doing that is CT scanning. And I'm going to talk a lot about CT scanning, so I just wanted to sort of show you what that looks like and how we use CT scans and reconstruct them in order to describe fossils. Now, we're quite lucky at the Evolutionary Studies Institute at Wits. We have our own micro CT scanner, which has proven to be an extremely important and useful tool. So if you think of a CT scanner, it works in a very similar way as your round-the-mill hospital X-ray, except that instead of just getting one picture of your bones, you get an entire stack of them. So if we go through it, so this is a specimen affectionately called the gun. It's BP5241, so it's a Massospondylus subadult that I scanned for my honours, so in 2013. And if we go through the stack of the CT scans, you'll start seeing All right, so all of these are cross sections. So each of them represents a slice going through the specimen. You can see in dark here, this dark gray stuff, that's actually rock that's still embedded in the specimen. And all of the white bits are individual bones. And what happens is I can then go and color in all of these individual bones, and these will get reconstructed in 3 dimensions. And it allows us to look at bones inside the skull or look at morphologies and structures inside specimens without having to manually prepare it, so manually remove all of this rock matrix that's around the specimen. And it's an extremely non-destructive tool that we can use when looking at specimens. So this gives us something that looks like, hopefully, this. So this is what it looks like once you've sort of colored in all of those individual bones. You can see that all of this matrix that was in the specimen, all of the rock, is now invisible, and you can peel all of these bones away and start looking at bones that you previously couldn't see. So these are all of the bones that surround the brain case of Massospondylus, that surround the brain. And you can also reconstruct morphologies that aren't preserved. So this is the inner ear of Massospondylus. So this normally would be filled with soft tissue. Obviously, the soft tissue doesn't fossilize, but what you can do using CT scans is you can essentially reconstruct the cast of this. And you can see, so this is the semicircular canals in the inner ears of the specimen, and they are contacting what's called the stapes, which is the middle ear. So we also have inner ears and middle ear. This is a very similar system going on here. I'm going to talk more about inner ears later on in the talk. Okay, so now we know who Massospondylus is. We know what tools we've used to best understand its morphology and describe it. What we haven't really discussed is why is Massospondylus such an important taxon. All right, and one of the reasons Massospondylus is extremely useful is that it is very, very common in the early Jurassic, about 200 million years ago. It's sort of the impala of the Jurassic, which means that we have hundreds of specimens. That doesn't happen a lot in the dinosaur fossil record. And not only do we have hundreds of specimens, we have a lot of skulls, and that's even more rare. And even better, we have skulls that range in size from teeny tiny little embryos, which you can see here, preserved in their eggs, to juveniles that fit in the palm of your hand, to slightly larger juveniles, to teenagers, which are about 20 centimeters long, to fully grown Massospondylus adults, where the skull is about 35 centimeters long. So Massospondylus is an extremely useful taxon to look at dinosaur growth. And you might ask me, but Kimmi, why do we want to know about dinosaur growth? Don't you worry, I will tell you. So dinosaur growth is an extremely important thing to understand for several reasons. I'm only going to go through a couple which are relevant to my talk today. And the first one is very simple, is that understanding growth or development helps us to better understand paleodiversity. So when we talk about paleodiversity, I'm talking about how many species were alive 200 million years ago. What did that ecosystem look like? And the way growth is related to that is that imagine you're a paleontologist and you find a very large skeleton of a dinosaur. You're going to describe it as dinosaur X, which is a very large dinosaur. Now you find a fossil of a much smaller dinosaur, and you're going to name it dinosaur Y, which is a much smaller species. So you have dinosaurs X and Y, which are different species. If it turns out that that little dinosaur is not actually a different species, it's version of dinosaur X. This changes the whole dynamic of your ecosystem and your diversity and your paleodiversity. Okay, so it's extremely important to understand how anatomy and morphologies change during growth so that we can better understand how many species we actually have that are present in the early Jurassic. Another reason, and this is a slightly more complicated, is that there are mechanisms involved in macroevolution, so very long-term evolution, that relate to growth of a species. And one of those mechanisms is what we call pedomorphosis, which is a big word, but it's fairly simple to explain. Basically, what pedomorphosis says is that you will get a derived animal or a derived taxon that has retained the juvenile features of its ancestor. And a very easy example of this is birds. So if we look at bird skulls today, there's research that has shown that bird skulls are pedomorphic. And that means that they have retained features and shapes that best resemble the juveniles of their ancestors. So if we look at T-Rex, here's mommy T-Rex and here's baby T-Rex. These are the ancestors of birds, and the baby T-Rex and the modern birds today have very similar skull shapes and skull features, and that's what we call pedomorphosis. So there is a relationship between growth and macroevolution. Okay, and that will come up a few times in my talk today. So we know about dinosaurs, we know about Massospondylus, we understand now why it's important to look at ontogeny, but why is it important to look at Massospondylus' ontogeny? And one of the main hypotheses that's been put forward about Massospondylus is that as a juvenile or as a hatchling, it walks on 4 legs. And then as it gets older, it shifts its posture and it shifts its gait, and it moves on to a more bipedal locomotion. Now this has fairly large implications, and this hypothesis was first put forward by Dr. Reisz et al. In 2005. I think Grant would have sent you guys the papers that I sent through, so a lot of the papers that I'll be talking about today are in that folder. And in this research by Dr. Reisz et al. In 2005, it's really a sort of pioneering work on Massospondylus ontogeny. One of the hypotheses that was put forward was this locomotory shift during Massospondylus' growth. And the reason behind that is, if we look at, so this is a plot from that paper, on the x-axis we have the length of the femur. So when we talk about the femur, it's the thigh bone. And then on the y-axis we have different skeletal elements. So these are vertebra, this is arm bones, the skull. And one of the main things that was noticed is that the humerus in green, the length of the humerus appeared to get proportionally shorter relative to the femur as Massospondylus grew. And that meant, the conclusion was that if the arm gets shorter as Massospondylus grows, then it shifts its posture. So when it's small, it has arms and legs that are about the same length, and as it gets older, its arms are shorter, so it must be going from quadrupedal to bipedal. This has fairly large implications on the macroevolution of posture in dinosaurs. If we look at sauropodomorphs, so that group of dinosaurs that includes Massospondylus, as well as these very, very large graviportal guys like Diplodocus and Brachiosaurus, there is a major shift in posture. So Massospondylus, adult Massospondylus, which are very basal and ancestral in the sauropodomorph lineage, are bipedal. They walk on 2 legs. And as we move through the evolution of sauropodomorphs, they adopt a more quadrupedal graviportal posture. Now, if there was a shift in posture during the growth of Massospondylus, so the ancestral state was to go from quadrupedal babies to bipedal adults, then it means that the derived sauropodomorphs could have evolved their quadrupedal posture through pedomorphosis. So again, they would have retained the juvenile features of their ancestors, in this case, quadrupedal locomotion. So this ontogenetic locomotory shift in Massospondylus has very large implications in the evolution of dinosaurs. And that's why it's quite important to better understand it. Now, although looking at limb lengths looks like a very simple way of doing it, determining posture in dinosaurs, adults and juveniles is not easy. There's many reasons for this. The main one is you need the sample, right? So if you want to look at whether there's a shift in posture between juveniles and adults, well, you need juvenile and adult fossils. That doesn't often happen in the dinosaur fossil record. Once you have the fossils, how do you determine posture? It's very difficult to do, and there's lots of methods that have been put forward to do this, some more successful than others. For example, this is a paper that came out last year. You could look at the center of mass of the animal, so you can model all sorts of different body proportions until the center of mass makes sense, and from that determine posture. But a lot of these methods that have been put forward require relatively complete skeletons, and again, this is very rare in the fossil record. So if your method requires you to have a complete fossil, a complete skeleton, in order to determine its posture, then you're going to reduce your sample size even more, because it doesn't often happen. Believe me, Jurassic Park and finding the entire skeleton, it's not common. I think I've been digging up dinosaurs for about 8 years, and I have yet to dig up a complete dinosaur. For some reason, they're always missing the head. So yes. And then the last bit is that most studies that look at posture in dinosaurs, well, because it's so difficult to do, we'll only look at one species at a time. If we want to get a broad picture, we need to use the same method on a bunch of different species so that we get sort of a better macroevolutionary idea of what's happening with posture. So one of the main aims of my PhD was a multidisciplinary approach to determine, or looking at postural shift in Massospondylus, and we did that using a variety of methods. So I'm going to go, the first half of my talk, I'll go through those methods. And the first one, which is also in the papers that I sent you, is a paper that came out with my buddy James Neenan, who's now in Oxford, in 2018. He's a fiend for inner ears. So inner ears look like this. If you guys remember in the spinning skull video, I showed you where they sit in the skull. And inner ears are really interesting and really important. I mean, we have semicircular canals in inner ears as well. All animals have them today. And they can tell us a lot about head posture. So your inner ear is not only responsible for your sense of hearing, it's also one of your main balance organs. And there's a lot of research that's come out that looks at the relationship between the geometry of your inner ears. So if we look at the inner ear, it's made up of 3 semicircular canals. So there's the anterior one, the posterior one, and the lateral one. And there's a lot of research that's come out on the shape of these canals and how that relates to posture in animals. And for example, in dinosaurs, it's been said that bipedal dinosaurs will have a much larger vertical semicircular canal. So these two, the anterior and the posterior ones. And there's also a relationship between the posture of your head. So this is the adult Massospondylus and the baby Massospondylus. And it's been said that in order for the head to be in alert posture, so the right posture, then this lateral semicircular canal needs to be held horizontal to the ground, which is how James has represented the skulls here. Okay, so if we have a relationship between inner ear shape and posture, and we hypothesize that our Massospondylus is changing its posture during growth, then we might expect to see a change in shape in the inner ear when we go from baby Massospondylus to adult Massospondylus. Now I'm not going to go in the specifics of these plots, but basically these are principal component analyses plots. The lighter dots are the baby Massospondylus, which you can see here, and then the darker dots are the adult Massospondylus. And these axes represent shape. So if we have animals that have a similar shape, then all of them should be plotting together. So for example, if we expect to have a change in inner ear shape from juveniles to adults, then we would expect to see the juveniles on one side of the graphs and then the adults on the other side of the graph, because there's that change in shape. We did not see or find that in the inner ear morphologies of Massospondylus. There was a slight variation in this part of the inner ear. And there's no research that shows that that's related to posture in any way. The main thing that we found actually was that the inner ear grows quite a lot, which had never been recorded in dinosaurs. So we don't have any evidence for a change in shape in the inner ears during growth, and therefore we don't have any evidence for a locomotory shift, at least in the inner ears. The second line of evidence was looking at limb robusticity. So this is based on a method that was put forward by McPhee et al. In 2018, when they described Ledumahadi mafubi. So Ledumahadi is, again, a proudly South African dinosaur from just off the border of Lesotho. It's very, very, very large. And the paper describing it was in 2018. And in this paper, there's a new method for inferring posture that was put forward. And it's quite a simple method, which is one of the reasons I really like it. But it makes sense. And the idea behind it is that if you walk on 4 legs, then you're going to distribute your mass evenly across your 4 legs. So you're going to be loading your arms and your legs in an even way. So in other words, your femur, which is your thigh bone, and your humerus, which is your forearm bone, would have similar thicknesses, right? Because they're responsible for a similar amount of weight. So that whereas if you walk on 2 legs, you would be loading and putting a lot more mass on your back legs than on your arms. So your femur would be thicker than your humeri. Okay, and that's what we mean when we talk about limb robusticity. It's the ratio between femur circumference and humeral circumference. And in this paper in 2018, they looked at a bunch of adult dinosaurs as well as hundreds of mammals alive today, both quadrupedal and bipedal, and they found that this simple ratio between humeral and femoral circumference is a really good proxy for determining posture in animals. And I thought, that's great, let's use that on baby dinosaurs. But before we can use it on baby dinosaurs, we need to make sure that it actually stays afloat and works with growth series of taxa that are alive today. So myself and my colleagues went around and measured a bunch of animals which are still alive today. So things like hippos, bears, cats, bipedal animals like kangaroos and jaguars and ostriches. And we looked at growth series. So for example, if we look at the ostrich here, the guys on the left-hand side of the plot are the babies, and then as we move to the right-hand side of the plot, those are the adults. And same thing for all of these lines. So each line represents the growth series of one animal. And if we look at humeral circumference, so the circumference of the arm versus the femoral circumference, or the circumference of the leg, you can clearly see that there's a split between bipedal animals, which are in orange, and quadrupedal animals, which are in blue. All right, and that seems to stay true for juveniles and adults. If animals, for example, ostriches today, they're bipedal as juveniles, and they remain bipedal as adults. So this looks like it's a really easy way of looking at posture in growth series of animals. And we also plotted humans, which are in grey. And interestingly, humans start off in the quadrupedal side of the graph and then shift into the bipedal side of the graph. So we seem to be able to detect those postural shifts. So now we've shown that it works with animals alive today. So the next step is to plot growth series of animals which are extinct. So we did it for several groups of dinosaurs. I'm only going to talk about the sauropodomorph dinosaurs, so the plant-eating dinosaurs we've been talking about. And if we look at, for example, this is Repetosaurus, which is a very large sauropod, one of the big guys, the graviportal guys. We've never expected a shift in it, and you can see the babies are here. They are plotting in the quadrupedal side of the graph, and as it grows, it remains in the quadrupedal side of the graph. So no shift for Repetosaurus. If we plot Massospondylus, so Massospondylus, the embryos are here, and here's the juveniles. It starts off as a biped, and it stays in the bipedal realm all the way through its growth. So it doesn't look like we have a locomotory shift in Massospondylus. We do, however, pick up a shift in this guy here called Massospondylus. Massospondylus is Massospondylus's cousin from Argentina. They're part of the same group. They're very, very similar and very closely related. And we don't see a locomotory shift in Massospondylus, but we do see one in Massospondylus. And this, again, has some implications for macroevolution. So this is a very, very simplified tree, phylogenetic tree of dinosaurs. I only want to focus on this bit down here because these are the plant-eating dinosaurs that we've been talking about. This is another paper that came out last year that also looked at this postural shift in Massospondylus, and they also found that it hatches, and as a juvenile it's quadrupedal, and then as a biped it's as an adult it's bipedal. Now, what is this? Why is this interesting, and why should we care about this postural shift? The main reason is if we look at where Massospondylus sits on the tree. So if you go from the left to the right, these guys here are much more basal sauropodomorphs, so they're the ancestral ones, and then as you move to the right-hand side, you've got the much more derived guys, so those big graviportal guys. Massospondylus here is in orange, so all the orange taxa are bipeds, and then all the blue ones are quadrupeds. If we look at Massospondylus, it's sitting in between, sort of in the middle of the tree. It's definitely a biped as an adult, but it seems to be sitting in a group of dinosaurs that's quadrupedal. And now that the exact relationships of Massospondylus are not completely understood yet. And there's two main hypotheses that have been put forward. So the fact that it's going from being a quadruped to a biped during its growth, and the fact that itself is a biped within a group of quadrupedal dinosaurs, that means that this group of quadrupedal dinosaurs would have had a common ancestor that was probably quadrupedal. So in this group of dinosaurs, you're going from a quadrupedal ancestor to Massospondylus, which is a biped. And Massospondylus itself individually is going from being quadrupedal to bipedal. And that's what we call recapitulation. So the growth of Massospondylus is actually recapitulating or mirroring its evolution. So going from quadrupedal to bipedal. That's another of those mechanisms, one of those links between development and evolution. And the other one, the other hypothesis, is pseudomorphosis, which we spoke about before. If Massaurus is going from quadrupedal to bipedal, and Massaurus is more basal than these very large graviportal sauropods, then it might mean that these very large guys, again, have evolved quadrupedalism through pseudomorphosis. So they have retained the juvenile features of their ancestor, which is Massaurus. Okay, so we don't have any evidence for a locomotory shift in Massospondylus using limb robusticity, but we do find some interesting stuff regarding Massaurus and the evolution of sauropod locomotion. All right, the last method that we use to look at whether or not there was a postural shift in Massospondylus is histology. Histology was a brand new world. I had never done histology before my PhD. It turns out it's incredibly interesting. And the way it works is you take a bone, you obviously get all the permits that are required and all the permissions that are required, because then you cut it up and you slice it and you put it on a slide and you look at it under the microscope. And basically you can look at tissue structures that obviously are not visible to the naked eye. And the stuff you can see is just phenomenal. You can see individual bone cells that, have been preserved for. Obviously the cells themselves aren't preserved, but you can see the shape and where they would have been 200 million years ago. And histology allows you to learn all sorts of different things about dinosaurs. One of them is how fast did it grow, as well as how old it was when it died. And it works in a very similar way to rings on a tree. If you look at this is the cross section of a basal sauropodomorph humerus, so arm bone. And you can see all these different lines going around it. And those are very similar to the lines that you get, or the rings that you get on a tree. Each line is what we call a lag, so a line of arrested growth. And basically, if you look at the distance between one lag and the next one, that represents the space between two seasons, which means that is basically equivalent to 1 year's worth of growth. So using these lags, we can estimate how old the dinosaur was when it died, as well as how fast did it grow each year. So if your dinosaur is going from being quadrupedal as a juvenile and then being bipedal as an adult, then you would expect to see some change in the growth between its femur and its humerus, right? Because at some point in time, its humerus is either going to have to start growing slower than its femur, or its femur is going to have to start growing much faster than its humerus. So we can use paleohistology to try and see if that's in fact what's happening. And from there, so if we don't see a change, then we don't expect to see a locomotory shift. If we do see a change, then there might be a locomotory shift. All right, so off we went. We cut up 26 Massospondylus individuals of different sizes. So we had juveniles, we had early subadults, we had late subadults, and we had adults. And in total, we cut up 46 elements, or 46 bones, individual bones. And these included all of the long bones, so the 3 bones of the leg and the 3 bones of the arm. And I'm only going to talk about the femur and the humerus today, because those are sort of the main ones. But basically, if we look at this plot here, on the x-axis, you've got the lag number, and the lag number is equivalent to the age. So this guy here is the embryo, and then as you move to the right, you're getting dinosaurs that are increasingly older. All right, so here you get to about 21 years of age. And then log circumference is the equivalent of body size or size. The circles are the femora and the triangles are the humeri. And the main thing that you can see is that as the dinosaur gets older, its humeri and its humerus and its femur have a very, very similar growth trajectory. There doesn't seem to be a major change. The humerus doesn't look like it's slowing down more rapidly than the femur. And what this tells us is we don't think that there's a locomotory shift that happens during the growth of Massospondylus. However, what we did find, which we weren't expecting to find, is that if we look at the spacing in these lags, so again, going from left to right, these are the smallest Massospondylus, and then as you move towards the right, these are the oldest Massospondylus. And then on the y-axis, we have the distance between the lags. All right, so imagine this is lag 1, this is lag 2, this is lag 3, this is lag 4. So what this is telling you is this is how much your dinosaur's grown in between year 1 and year 2. This is how much it's grown between year 2 and year 3, etc. And something that should be quite striking here is that all of these distances are very variable. There's no pattern. Normally what you would expect is juveniles should be growing very, very fast at the beginning, so the lag spaces should be very big, like you have here. And then as they get older, these lag spacings decrease, right, until they reach full size. That's not what we see at all. In fact, we have an extreme variability in lag spacing, and that's what we call developmental plasticity. And what that means is that the growth of Massospondylus is not necessarily regulated by its physiology, if I can say. It's mostly regulated, or it's affected by its external environment. So if the environment was good and Massospondylus was happy, it was growing really well. If the environment is bad, it's growing much slower. And one of the hypotheses behind this is that if we remember Massospondylus is alive in the early Jurassic, that's a post-extinction environment, and this might be a coping mechanism to living and recovering in a post-extinction environment. But we're still working on this. This is not published yet, so stay tuned. Okay, so that brings us to the end of the first half of the talk, which is basically Massospondylus carinatus did not go through any locomotory shifts during its ontogeny, and juveniles would have been bipedal. Now, as I said, Massospondylus is incredibly useful for looking at developmental growth in dinosaurs, and one of the data points in the Massospondylus growth series is basically the ideal data point because it is about as early as you can go, and those are the embryos. So we do have a clutch of eggs of Massospondylus that preserves embryos in them, and that's going to be the second half of my talk, which is sort of the work that we did on that. So a bit more about the background on those eggs. So the eggs were discovered in 1976 by Professor James Kitching, who is sort of one of the very famous fossil hunters in South Africa. They were found in Golden Gate National Park, right next to Clarence. So if any of you ever go to Clarence, make sure you stop in Golden Gate, because actually, so this is the site where they were found. It's right on the side of the road. They were actually found when the road was being cut up. There is a museum being built about here, which myself and Professor Jonah Chouanier are the scientific advisors for, and it should be opening early next year. And it's a whole museum dedicated to Massospondylus and dinosaurs. So make sure that you guys go and visit it next year. So they were found in 1976, but the eggs were actually only published for the first time in 1979. Very little was said about them. It was just said that they were eggs. And 7 of them, and and that they did contain embryonic material. And it's really between 2005 and 2012 that the research on these embryos really started happening, and all of it by Dr. Reisz and colleagues. And in order to do this, the eggs had to be prepared. So when we say prepared, we mean that we're removing all of the rock from the bones. And so this was done in the early 2000s. And it revealed 2 embryos. So here's a complete one in side view. You can see here's the skull, here's the back, here's the leg, here's the arm. So it's curled up in the fetal position in its egg. And then there's a second embryo here, but this time you're looking at it from the top. And you can't really see, there's its neck going into the egg. You can't see much more than that. So these embryos were described based on the surface material in between 2005 and 2012 was when the research happened. And although, I mean, these eggs are absolutely incredible. I mean, look at how cute they are. And dinosaur eggs are very rare. The Massospondylus dinosaur eggs are not the only dinosaur eggs that we have in the world. There have been other dinosaur eggs found, notably a lot in China, in Portugal, I mean, all over the world. And there's a lot of research that's been done on dinosaur embryos. For example, if you look at embryonic tooth microstructure, so if you look at the histology of the teeth themselves, you can determine incubation period in these dinosaurs. You can also look at the eggshell microstructure and start looking at soft eggs versus hard eggs, etc. One thing that hadn't really been done is looking at the pattern or the order in which the bones of the skull in dinosaur embryos appear. So all embryos, as they form, they're not magically a skeleton that just appears. The skeleton appears bit by bit as the bones ossify. And the order in which this happens is basically, we didn't know the order in which that happened in dinosaurs yet. And that's one of the main reasons why we went to France and we scanned these eggs. So we tried to CT scan them at Wits. Because they're so small, basically all you got was specks of white, and there was nothing you could do. So we put in an application to go to the synchrotron, so the European Synchrotron Radiation Facility in Grenoble in France. And we're very lucky because South Africa has a partnership with the ESRF, and so that allowed us to take the eggs there. And basically the way a synchrotron works, it's pretty cool. It's like the bazooka of CT scanners. You have a ring, so this ring is about 1 kilometer in circumference. And the way it works is you have a thing called the electron gun, which shoots out electrons, and they speed up in this little ring very, very, very fast until almost the speed of light. And then they get stored in this much, much larger ring. And then as they speed up, they emit X-ray beams. They then get sent into these individual tunnels that come off of this big ring, which are beam lines. And this is what it looks like inside one of the hutches of these beam lines. And it works in a very similar way to a CT scanner. It's just, much, much, much more powerful. So the beam line or the X-rays come out of here, you put your specimen here, and then your detector's there. So this is what captures the images. So we put our eggs over here and scan the specimens and managed to get incredible resolution on the scans. And that then allowed us to reconstruct all of these embryos. So I'm only going to show the skulls. So for example, this is the same skull that you have here, but you can see all the different colors of the individual bones. So I'm only showing you one side. This is the same skull that you had here. Of course, now what happens is we can look at the underside of the skulls. And then we had a little Kinder Surprise egg over here because this one also contained embryonic material, which we didn't know about beforehand. Obviously, Murphy's Law, you can see it's cracked, and it's cracked right where the skull is. So we only had a few cranial bones, but it's still data that we can add. Now you can see these skulls have been squished. So the one's been squished mediolaterally, so side to side, and the other one has been squished top to bottom. So then you take all of these individual skull bones, you put them back together, and you get a nice 3D reconstruction of what your embryonic skull would have looked like. Now this is actually teeny tiny. It's only about 2 centimeters long. So these bones are actually very, very, very small. Okay, so now we have our dinosaur skull. What we need to do is compare it to animals that are alive today in order to determine if its order, its ossification sequence in the egg is similar to its relatives. So the animals we chose to look at are what we call saurians. So saurians is this very large group of animals that includes from lizards all the way to birds. And we sampled lizards from the literature. So we had bearded dragons. Then we had scans of turtles, crocodiles. We have Massospondylus, which is the sauropodomorph. And we had birds. We had chickens. And for the turtles, the crocs, and the birds, we got data from the synchrotron where they had already scanned all of these crocodile, chicken, and turtle eggs at different stages in the incubation period. And then basically what we did is you look at the crocodile at 39 days in its incubation period, and you say it's got this skull bone ossified, this skull bone ossified, this one's missing. You go to 48 days, it's got this skull bone, this one's missing, etc. until you get a pattern. And you can tell which bones ossify first, as well as how ossified they are at which stage. And then you can compare your Massospondylus. So the first thing is that we found that turtles, chickens, crocs, and lizards have very similar sequences in which they ossify. And that then allows you to take your Massospondylus embryos, compare it to here's the chicken, here's the crocodile, here's the turtle. cross-reference it and say, okay, if we have these bones preserved or ossified, and these bones are missing, then it must be at X% in the developmental sequence, or it must be at X% through the incubation period. And what we found is that the level of ossification in Massospondylus is more similar, or most similar, to the Saurian relatives at about 60% through the incubation period. That was the main finding. So it's a lot younger than we previously thought. It was previously thought that they were right near hatching, so right near the end of their incubation period, and we found that they're very, very young. And the second thing we found, which is very interesting, is that it has two types of teeth. So these teeth here, you can see they're tiny. They're about 0.5 millimeter across, so it's more than the tip of a toothpick. These teeth here have large serrations, similar to the adults, and these ones are much more simple conical teeth. And we hypothesize that these guys here are what we call null generation teeth. So crocodiles and geckos and lots of reptiles today will develop teeth while they're still in the egg, called null generation teeth. They will then reabsorb those teeth, or they'll lose them. And then after that, they'll get their functional teeth, which are the teeth that they'll hatch with. So we have what we think are null generation teeth in Massospondylus. And that brings me to the end of my talk. So just if you want to know what I'm working on now. So basically on the eggs, the main bits are now looking at the rest of the skeleton, looking at the teeth histology to try and determine incubation period. And then we're also trying to learn a bit more about the eggshell microstructure, not just on these eggs, but on other eggs from the same area. So with that, I would like to thank all of my funding bodies for supporting me over the past few years. And then that's all the Massospondylus for now, if you guys have any questions.
Kimmi, thank you so much. That was absolutely incredibly fascinating. Really, really interesting. Thank you. And just showing how much we can integrate some really cool technology with paleontology. So we have a whole bunch of questions coming through if you're ready to answer some, Kim. So keep your slides up. Sure. Let's start with a question from Mark, which I'm publishing now. And Mark, I'm not sure if you can see them, Kim, but I'll repeat them because the YouTube audience later won't be able to see them. So, did you physically cut up the bone for histology? Could CT scans give you the same information without cutting up the specimen?
Okay, so that's a very good question. Yes, I did physically cut up the bones for histology. I did get all the permits. So one of the things that you usually do in histology, or at least what we try to do, is obviously you're not going to cut up all of the bones in a specimen. You select specimens where you have both humeri preserved, and you cut up usually the ones that are not as well preserved or are incomplete. There is a lot of work that's being done, especially now, on using synchrotron scanning on histology. They are getting very promising results. They are, however, some things that you can't see using CT scanning, so they answer different things. The advantage of using synchrotron on histology is that you get a 3D visualizations of all of these microstructures. The disadvantage is that there are certain structures that you can't see. For example, this stuff in cross-polarized light is much more difficult to see. It's also very difficult to get the resolution high enough. So synchrotron scanning is the way to go, but to get an entire cross-section like that is quite difficult.
Okay, great. Then the next question from a user named ProSauropodFan409. Is there any change in Massospondylus growth patterns as individuals get further out from the Triassic-Jurassic extinction and ecosystems presumably stabilize?
That is also an excellent question. So this is one of the reasons why the histology specifically hasn't been published yet, is that we're now trying to focus on comparing the Massospondylus from the end Triassic to the ones from the early Jurassic to the ones from the late Jurassic. At the moment, we can't see any patterns, but we need to sample sort of more focused on those different time periods.
Okay, here is a question from Matthew Carano. What are your thoughts on null generation teeth? Do they serve some embryonic function? Are they a relic of something evolutionarily prior?
It's cool that Matt Karan is watching my talk. So with regards to the embryonic teeth, that's also an excellent question. It's actually something that I've been discussing with my co-authors quite a bit. So one of the ideas that my colleague Vincent Fernandez put forward, which is I think quite an interesting one, is so the fact that if we look at the embryonic teeth, so there's null generation teeth and first generation teeth, the functional teeth, at the same time, but not in the same tooth position. And you don't need a null generation teeth present in order to get a first generation teeth. But what Vincent was saying or hypothesizing is that what might happen is that in order to get, because dinosaurs replace their teeth constantly throughout their lives, it's possible that in order to have a functioning replacement pattern to start, then it needs to start very, very early. So they start with those null generation teeth, which are really, really easy to make, and then replace them very quickly. And that sort of kickstarts that replacement sequence. Another thing might be sort of a more deep-rooted ancestral remnant that we don't know about yet.
Okay, great. Thanks, Kimmi. Right, the next question is from Mark van der Brunt. And now I'm going to be challenged into getting my pronunciation of the species correct. Excellent talk, first of all. There is one Pariasaur, Bunostegos, that has a very upright forelimb posture. All other stocky Pariasaurs are sprawling and effectively permanently doing push-ups, even more than I'm doing in lockdown. How would you test the forelimb posture of quadrupedal Pariasaurs?
So that's also a very good question. Is how does limb loading change between sprawling quadrupeds and erect quadrupeds? That hasn't. I don't think that's been done yet. I know that it was in the works in the next few years to try and look at, so using the same limb robusticity method that we use, we wanted to focus sort of on saurians as a whole. And look at exactly that. So is there a change in limb loading and limb robusticity between sprawling gaits and erect gaits? But I don't think it's been done. Why don't you go measure your Pariasaurs, Mark? We can then tell.
Okay, Kim, are you still okay to answer a few more questions?
Yeah, go for it.
With the audience, good. Right, so here's another one from Anonymous. What would the advantage or reason be behind a locomotor shift during ontogeny?
So that's also, I mean, this is a debate I was having today. So what is the advantage with using, so the ontogenetic shift might be how can I explain this? There must be a link somewhere between macroevolutionary patterns and growth patterns, or if there is a link between the two. It's hard to tell if it's more advantageous to have an ontogenetic shift or a macroevolutionary shift. So I was discussing this morning, is animals that adopt quadrupedal posture through pedomorphosis. Why? What is the benefit of using pedomorphosis to adopt quadrupedal posture? And I think it's not necessarily the benefit of using pedomorphosis to adopt quadrupedal posture. It's the fact that locomotory shift and that trend is already present. And for example, in sauropods, if you want to reach very large body sizes, then quadrupedalism is the way to go. And that shift from bipedalism to quadrupedalism is a trend that's already existing. So it might be that there are some constraints in patterns. Ontogenetically, that's a very good question. I mean, then you start asking what's the advantage of being a bipedal versus a quadruped?
Okay, great. Here's another question from Javier. What was the environment like in Massospondylus' time?
So late Triassic, early Jurassic. So that's Upper Elliott, Lower Clarence. So it's getting much. So it's already quite a dry environment. So the Elliott Formation is mainly mudstones. So it's a lot of floodplains and riverbanks. And then as you start going into the Clarence, it dries up quite drastically until you get to the Clans Formation, which is basically an aeolian desert.
Okay, here is another question, which starts off by saying wonderful talk, which I certainly agree with. Are there important changes in the teeth of Massospondylus throughout ontogeny? And that's from Diego Paul.
None, no. So if we look at the tooth morphology of, for example, the juvenile that I showed at the beginning of the talk, I have to go quite far back. All right, so this guy over here, for example, its tooth morphology is basically exactly the same as this guy here, which is the gun, and it's almost, I mean, it's definitely exactly the same as the larger specimen, Big Mama. So no, we don't see any morphological changes in tooth morphology.
All right, so here's another question. Thank you so much for all of your questions. You're getting quite a peppering here, Kimberly.
All right.
You talked about a bit about the need to understand the past to understand where we are going. What does our knowledge of these dinosaurs add to understanding of how species might react to future environmental instability?
Oh, that's a big question.
Indeed.
All right.
Thank you, Matt and Emily, for that question.
Oh, I know Matt and Emily as well. I guess understanding. So if we look at, so there's several bits to that actually. So looking at Massospondylus as an individual species and looking at, for example, this developmental plasticity, which may or may not be a coping mechanism to the environment, can help us understand, for example, or predict which animals we think might be able to cope best with drastic changes in environment in the future. Secondly, and this is sort of a zooming out idea, is how does diversity and how do sort of populations recover after an extinction event? So if I mean, there's a whole bunch of people who work just on this, so looking at rates of extinction, diversification after extinction, and I think this is something that's being learned about on a daily basis, and they're very, very complex models to do this. So yeah, so understanding one, rates of extinction, so are the extinctions that happen in the past, how do they compare to the extinction that we are undergoing now, that we might undergo in the future, as well as how can we expect diversity to recover from that, and what might happen with diversity after that. So the level of diversification after that.
Okay, and we'll end off with one last question from Anonymous, and that is, can we expect ontogenetic shifts in other groups of dinosaurs that shifted evolutionarily from bipeds to quadrupeds, like ceratopsians and hadrosaurs?
A very good question. Yeah, so if we go back to this tree here somewhere, yeah. So for example, here are the ornithischians, and we have ceratopsians within ornithischians, obviously. And for example, Psittacosaurus undergoes an ontogenetic shift, but not from quadrupedalism to bipedalism. It's the other way around. Sorry, no, I got that wrong. Don't quote me on that. It does undergo a locomotory shift, but not necessarily the other way around. Yes. So yes, we do see locomotory shifts in other species. Okay.
Well, Kimberly, I can just thank you for a really fascinating talk. I think the size of the audience indicated how interesting it was. We had about 50 people, so we hope that you all join us again next week. And I'll just very briefly put up next week's talk so you can all see that. And we will do that shortly. You should be able to see it now. The talk for next week is by Ray Durheim and Musa Manzi from the School of Geoscience at Wits University. And this is a talk focusing on seismic imaging of a 2014 magnitude 5.5 earthquake rupture beneath the Moab Khoesong gold mine in South Africa. So we hope that you'll join us for that really interesting talk that shifts us from paleontology straight to geophysics and subsurface mining. And thank you once again for joining.